Sure, it doesn't even go after
the second.
all of these today.
I'll see what I can do.
Couldn't figure the ag out.
Okay, yes, the question is, we will have lab tomorrow because you will get your lab exams back, and I'm going to go over those. I've got pictures of the results and things, so I want to go through those. That won't take the whole period, and so the remainder of the time is available for you. You can do with what you want to. There are things you obviously need to work on. And that will be the case next Wednesday. Wednesday is your time. Friday, I'm going to ask you to all be here because we'll do a checkout, and I'm going to have you kind of reorganize your lab drawers to make sure everything's stocked up in there. But it's a matter now of finishing up projects and working on unknown. Okay.
So when you say things are really last day of class, is that Friday or Thursday?
Friday. Yep, I said Friday, I think at 4 o'clock, right? We're in the home stretch. Okay, today we're ready to do something that we've been building up for quite some time now, and that is talking about the interactions that take place in the third line of defense. But before we do that, let's step back for just a second and look at all three lines of defense. What's the primary purpose of your first line of defense? Yeah, prevention. Okay, trying to prevent infection from occurring in the first place. And so there are a variety of physical barriers, tissues, some biochemicals, some processes like peristalsis, for example. But it's pretty much all about prevention. Second line of defense. What are we doing? Well, okay, more than likely for the second line to be activated, there has been a breach of the first line of defense. So now it's about responding. And the second line of defense responds pretty rapidly. So this is a fairly rapid response. It is a pretty generalized response in that several different pathogens can initiate the same general response against infection. It involves things like inflammation, activation of complement, production of interferon, and phagocytosis. And what's the goal here? The goal here is to immediately respond to things that might be threatening. Okay, in the case of inflammation, a mechanical injury can cause inflammation. That has nothing to do with infection necessarily. But this is just kind of a general response to things that might be potentially harmful to you. And a lot of times what the purpose of this is basically to buy some time. To buy some time for what? To get that third line of defense kicked into play. Third line of defense is highly specific. It is a phenomena that you acquire as a result of experience being exposed to pathogens. Remember that line 1 and line 2 are considered to be innate, things you're born with. Line 3 is something that you acquire during your lifetime. But it is a slow response. Okay? And that's why having this second line of defense, kind of reinforcing it, is really important because it takes time for this to particularly develop, particularly if the exposure is your first exposure to the threat, if it's a primary response. Because remember, there's that latency. You only get a limited type of antibody produced, IgM. So it's really when you have become educated and developed some memory that this is really a powerful response. Okay, third line of defense consists of two overlapping parts or arms that we talked about. And it's covering kind of the gamut of scenarios. One scenario is you've got an infectious agent in your tissues and it's circulating around in your blood, or it might be in your lymphatic system circulating around in the lymph, or it might be in that extracellular fluid. This could be toxins, they could be virus that have yet to infect cells, they could be bacteria that have gained entrance. The second part, and the response to this is your cell-mediated immunity. So what happens is once cells become infected, we get this response initiated that eliminates those infected cells, virus-infected cells or cancerous cells. At the same time, we've got a humoral immune response working, which is geared toward production of antibody, and that is dealing with these antigens kind of before they gain entrance to the cells. So we've got the two kind of scenarios going on, and part of the immune system dealing with both of those scenarios. So what we're going to do is we're going to take a look first at the cell-mediated immunity. How do we kill damaged host cells, cancerous, virus-infected cells, and then look at humoral immunity and see how antibodies are produced, and we now have a good sense, hopefully, of what producing an antibody does for you in terms of your overall defensive processes. Cell-mediated immunity is more of a T-cell mediated process. So the players we're going to be talking about are T cytotoxic cells, T helper cells, and the humoral immunity is mainly a B-mediated response, because it's the B cells that produce the antibody. But in most cases, this response needs the assistance of helper cells as well, the T helper cells. So, cell-mediated immunity. What we're going to concentrate on when we look at this are kind of three things. What are the cell types that are involved? Okay, what are the major cellular players? What are the major receptors involved? Because those cells have to be able to recognize one another. And then what are the chemical signaling molecules involved? So when we look at some of these diagrams in a minute, they're going to look pretty confusing. But if you kind of start trying to concentrate on this and break it down this particular way, going, okay, what kind of cell is that? What kind of cell is that? What receptors does it have? That pretty much defines what the cell type is. T helper cells have different receptors than T cytotoxic cells, for example. And then what are the cytokines that are involved? Because cytokines have affinity for cell types, because cell types have different receptors. So if you just concentrate on those things, it should be helpful. So the major players for cell-mediated immunity are some kind of antigen-presenting cell. Could be a macrophage, it could be a dendritic cell. What determines which type of antigen-presenting cell is involved? A lot of things, but one of the big things is, how did that antigen get introduced in the body in the first place? Was it ingested? Was it injected? That's going to kind of help to determine what type of antigen-presenting cell it encounters. Okay? T cytotoxic cells or T killer cells are the cells that really do the killing themselves, and we'll see how they do that. But then also there's going to be a cooperation that occurs between the T cytotoxic cell and a type of T helper cell called a T helper 1. So one of the associations that you should make is an association between T helper 1 and cell-mediated immunity. Later on, we're going to see that there's a second type of T helper cell, T helper 2s. They're going to be associated with humoral immunity. So these are some of the kind of associations you should start forming in your mind if you're going to wrap your head around this. Purpose, kill damaged host cells. What are the cells that do that killing? They're the T cytotoxic cells. But the question is, what turns them on? That's where the cooperation between T helpers and antigen-presenting cells are going to be important. Why? Because the antigen-presenting cell is going to show the antigen to those killer cells and say, here it is. Here is what you need to be responding to. What role does the T helper cell play into? particular phenomenon. It secretes some cytokines that are necessary for the full activation of those killer cells. Okay, so here's just a chart that just kind of summarizes the subset of cells that are primarily involved in cell-mediated immunity. T helper 1s. Oh, I'm sorry, these are subset of all T cells, sorry. So, T helper 1s, T helper 2s, T cytotoxic cells. Cell-mediated immunity primarily, humoral immunity. Cell-mediated immunity. So we're going to start looking first at kind of this relationship between T helper cells, T cytotoxic cells, and then what's not showing on here because they're not T cells are the antigen-presenting cells. This shows you the primary receptor types on these particular cells. And then this shows you what some of their important functions are. So, what are the major steps involved in the activation of a T cytotoxic cell? What starts turning one of your T cytotoxic cells into a killer that is going to attack your cells? Well, they've got to be shown an antigen. That antigen actually is first shown to a T helper cell and a T cytotoxic cell simultaneously. I'll show you a picture of that in just a second. Those T helper cells and T cytotoxic cells have to be activated. They have to get the right signals. Part of that signal involves being shown an antigen. Another signal that's involved is being exposed to the correct cytokine. What does that do? That starts the T cytotoxic cells to start dividing. That's basically what the term clonal expansion refers to, is a cell starts dividing and forming identical copies of itself. So that T cytotoxic cell has been sitting in your body dormant because it's never encountered its antigen. But under the right circumstances, if it becomes activated, that particular cell now starts dividing and forming a whole army of itself that can then respond to that particular threat that it's now detected. T cytotoxic cells are kind of interesting because they need initial signals to get going. But once they start dividing, they start to stimulate themselves to divide more. This is a great example of a positive feedback loop. Remember that I said that in immune reactions, positive feedback and negative feedback both are important. Negative feedback tends to shut the system down when it's time. Positive feedback builds up the response and gets it going like that rock down the hill. Just through this particular picture in here, because we're going to see a reference to dendritic cells in just a minute in several of the diagrams. So this is a very common antigen-presenting cell that's going to be interacting with T cells to get the cell-mediated immune response. Remember that these are phagocytic cells, and that they have the capability of not only phagocytizing, but then processing and presenting antigen to other cells. So they're antigen-presenting cells. And then also secreting various cytokines which stimulate those cells. Kind of think of them in contrast to neutrophils, which are just pretty much phagocytic. They chew up and spit out. They don't present antigen. They don't secrete any cytokines to speak of. Okay, so here's a diagram we need to spend a little bit of time looking at. Up at the top, we have this cell labeled DC. That stands for dendritic cell. So that is your antigen-presenting cell. Remember that all cells in your body are characterized by having MHC1 antigens on their surface, receptors on their surfaces. And those MHCs are displaying any internal protein that that cell normally makes. But in the case of this dendritic cell, any antigen that it is ingested from outside. So MHC1s can display both endogenous, their own protein antigens, which the immune system should ignore, but also exogenous antigens, things that it is phagocytized that are foreign. And that's really what we're interested in, is those exogenous antigens that it's displaying on its MHC1s. Additionally, however, dendritic cells, because they're antigen presenting, have MHC2s. Remember that MHC2s are restricted to a certain subset of cells in your body. T helper cells, B lymphocytes, macrophages, and dendritic cells. The major third line of defense immune players. Okay, so this is a pretty complicated cell. So, what's going on right here? Okay, this has ingested something foreign, broken it up, and then inserted pieces of that foreign substance on both its MHC1s and its MHC2s. And it's now going to present those antigens to different cells. The two cells that it's going to present it to are inactive T cytotoxic cells. They're sitting in your lymph nodes waiting. And inactive T helper cells. They're sitting in the lymph nodes waiting as well. Now, here's something that you need to remember. T helper cells are MHC2 restricted. T cytotoxic cells are MHC1 restricted. So what does that mean? That means that they can only respond to, or if you want to use the term see, the antigen, if it is associated with an MHC receptor. If that antigen is just floating around free in the body fluid, those T cells won't respond to it. They won't see it. So, this particular T helper cell right here is inactive. It's never seen antigen before. The antigen presenting cell now shows it that particular antigen. And it recognizes it because the antigen epitope that's referred to as here is complexed with its MHC2 receptor. Aha, I see it. There it is. Okay? When that receptor binding takes place, that turns on this antigen presenting cell to start producing a cytokine that it hasn't been produced before. I'm only going to ask you to remember 3 cytokines. Okay, the 3 cytokines that I'm really going to be interested in, and I've got a little diagram later on, are interleukin 2, interleukin 4, and interleukin 12. So here's interleukin 12. It's being produced by the antigen presenting cell, and it's affecting the T helper cell. We didn't produce interleukin 12 until these 2 cells hooked up. So, what does this interleukin 12 do? It takes this dormant cell and it now stimulates it to start differentiating. And so this immature T helper cell, a lot of times it's illustrated in diagrams as T helper naught, T helper 0. If it gets the interleukin 12 signal from the antigen presenting cell, then that's going to send it down this pathway of differentiation. And it's going to start turning into this T helper 1 cell. What do T helper 1s do then? Once they've differentiated into a T helper 1, they start producing their own cytokine, interleukin 2. And what does interleukin-2 do? It affects this T cytotoxic cell. It starts helping to activate it. The T helper cell has receptors for interleukin-2. So it's going to respond to that particular cytokine. So that's what's going on over here. But at the same time, this antigen-presenting cell is interacting with another subset of cells, the cytotoxic cells. So you've got this inactive cytotoxic cell. It's never seen its foreign antigen. It's just sitting there waiting and waiting and waiting. It's not responding because there's nothing to respond to. But now, the antigen-presenting cell shows its ingested antigen to the T cytotoxic cell. The antigen is complexed with an MHC1 receptor. T cytotoxic cells are MHC1 restricted, so they can see the antigen. It's in the right framework, it's in the right context. And once this pairing occurs, this inactive T cell starts making interleukin-2 receptors. It wasn't making those receptors before. So it's getting a signal by this binding that basically says to it, hey, let's express some new genes. Let's start making a new receptor that's going to respond to a cytokine, interleukin-2. This shows you some of the detail of the interaction that's taking place right here. And the only reason that I like to point this out is it shows you the involvement of some other receptors besides the MHC1, the T cell receptor. For example, this shows you the involvement of the CD8 receptor as well. Remember, T cytotoxic cells are defined as being CD8 cells. That's the CD receptor they have on their surface. T helper cells have a CD4 receptor, which they're not showing here. Okay. There's some insight into kind of the receptor complexity and matching that has to occur. Okay, so now because it's been activated by being shown the antigen, this now is expressing some receptors that can respond to the interleukins that this activated T helper cell is producing. You see how we don't get to here until the antigen presenting cell activates these cells and activates these cells? And then ultimately what we do is we now have a cell with receptors that can respond to a cytokine. What does that interleukin-2 do? It starts driving now the development of those T cytotoxic cells. And they start dividing. And we form some memory cells, and we form some effector cells. The effector cells are the actual killers. Now, here's where it gets a little bit weird, but kind of interesting too. Remember that these have interleukin-2 receptors. Where were they getting their interleukin-2 previously? From this activated T helper cell. But once they start differentiating, they start producing their own interleukin-2, which reacts with their own receptors, which causes them to produce more interleukin-2. So we get this positive feedback here, this self-stimulation, and that basically just makes things divide even faster. So now we've got this whole battery of identical T killer cells, which are ready to attack tissue that has become infected with this particular antigen that was presented in the first place. So if that were a virus-infected cell, this population of T killer cells would start seeking out all of your cells in your body that were infected by that particular virus. If it's a cancerous tissue, then it's going to start seeking out all of those cancer cells and destroying them. Should it destroy normal cells? No, because those T cytotoxic cells that would have responded to normal host antigen should have been eliminated during the development of the immune response, during that clonal deletion process. So we really only should be left with these inactive cells that would respond to foreign. Now they've been shown foreign. And now they're ready to respond then to cells that contain that foreign substance. That's one of the really interesting issues about cancer is if your third line of defense works against cancer, why do we get tumors? What allows cancerous cells to occasionally escape detection by these T cytotoxic cells, and therefore they can start to proliferate and grow? And the answer is we don't know many times what causes that. But one of the things that is important is that some of the cancer therapies that are being really effective right now are therapies intended to try to boost your own immune response against those cancerous cells. So can we give people infusions of interleukins, for example, which then might beef up their entire immune response? And now they might attack the cancer where they previously didn't. And so the fusion between immunology and cancer research is becoming very, very strong. So pretty complicated, right? And this is just kind of scratching the surface, because remember that I said that we've identified to date about 35 interleukins, and there are a bunch of other CD receptors that have been identified. In a couple of other diagrams, you'll see, I'm not sure why they put them in there, other receptors shown, which we haven't even talked about yet. So this is really kind of bare bones to give you an idea of the overall picture of what's happening in cell-mediated immunity. But the key is that you should have populations of both immature T helper knots and T cytotoxic cells sitting in your immune system locations, like the lymph nodes or the spleen. Sitting there waiting. Each of them having a unique T cell receptor which will react to a particular foreign antigen. And then if they are shown by the antigen presenting cell, those particular antigens in the right context, MHC2 context here, MHC1 context here, things start happening. And cells start growing and differentiating and dividing. And cytokines start being produced. And cytokines bind to receptors, which cause additional changes in those cells. And typically the changes that occur are changes in gene expression. And those cells now start making new proteins they didn't make before. And those proteins might be cytokines, they might be factors that trigger differentiation down cellular pathways. But the real key is to start turning on some genes that were dormant. Eventually, we produce these effector cells that do something, and what they're doing in this particular case is destroying your body cells that have become damaged and are expendable. Okay, how does T killer cells actually kill an infected cell? Okay, so once we form these populations of cells, what's happening? Well, they've got to bind to the infected cell. So again, there is some receptor binding that takes place here. Over here to the left is an MHC1 receptor that has a foreign antigen associated with it. And the T cell receptor on the cytotoxic cell recognizes that and binds to it. They're not showing over here for some reason the CD receptors that they showed in the previous diagram. I think they're just trying to keep it simple. That particular binding initiates some changes in the T cytotoxic cell. There are some vesicles that are present in the T cytotoxic cell. One type of vesicle contains a molecule called perforin. Another type of vesicle contains a different type of molecule, a protein, called a granzyme. So when this infected cell, in this case a virus-infected cell, is bound by a T cytotoxic cell, How does this cytotoxic cell know to bind to this cell? This receptor interaction right here. Okay, it sees the foreign antigen on the surface and binds to it. It initiates those vesicles to start fusing to the membrane of the cytotoxic cell and releasing their substances that were inside those vesicles. Perforin is a protein that's released, and it starts to polymerize on the surface of the infected cell. And it forms a pore complex. What does this look similar to that you've seen before? A bunch of proteins polymerizing on the surface of a cell and making a pore in the surface of that cell. Isn't that what happens when you form the attack complex and complement system? All those proteins polymerize on the surface and make that pore. What happens in complement? The pore allows the cell to lyse. What's happening here, however, is that pore complex is an entry route for this other type of substance, perforin. And granzyme. Granzyme then enters that pore complex and gets inside the virally infected cell. What does granzyme do? There are some cells in the cytoplasm of your normal cells called apoptotic enzymes. They induce cell apoptosis, cell destruction. But they're inactive. But in the presence of granzyme, they become activated. And once those enzymes become activated, apoptosis is initiated, and this virally infected cell starts to shrivel and die, and is killed. And all this is doing right over here is showing you some of the other types of receptors that we haven't talked about that are involved in this particular process as well. You can pretty much just disregard this particular one because we haven't talked about CD95L and CD95. Really, this is the key over here. Net result is this virally infected cell is destroyed. And if you can destroy that cell before the virus has completed its life cycle, the cell's not going to lyse and produce new virus. If this is a cancer cell, then that cancer cell will no longer divide and contribute to the formation of a tumor. So a lot of things have to happen before these cells become activated so they can then do this. You don't want to do this lightly. You don't want to destroy your own cells if there's not a real reason to do it. So you want to really make sure that these particular cells are bad guys, that they have been infected. Okay? Here we get the proper recognition if this cell has been activated for them to detect the cell that has been infected. This epitope right up here is the result of being infected. This is not a normal cellular protein that's being displayed right here. This is a virus protein that has been displayed because that cell has been infected by a virus or has become a cancerous type cell. Okay. Unfortunately, that is the more straightforward of the two systems. Cell-mediated immunity. Humoral immunity is a little more complicated, but there are some of the very same kind of concepts working with here. So, what's going on here? Well, the goal of humoral immunity is to produce antibodies, as opposed to killing infected cells. And again, hopefully you have a better understanding now for why it's important for antibody to be produced. It can be an opsonin, stimulant complement, etc. So what kind of interactions are taking place here? Well, we've still got antigen-presenting cells involved. We still will have immature T helper not cells involved. which hopefully will differentiate into T helper 2 cells. And then we've got the immature dormant B lymphocytes involved as well. So the result of these cellular interactions is to turn these on. So they start forming plasma cells, and the plasma cells start secreting antibody. Okay, when we talked about antigens, I said that there were several different types of antigens, and one of the designation for antigen types that I gave you was some antigens are T dependent and some antigens are T independent. Both of these have to do with humoral response. And secretion of antibody. T-independent antigens will stimulate B lymphocytes to produce antibody without a helper cell being involved. T-dependent antigens will only stimulate B cells to produce antibody if a T helper is involved. Okay. Here, T helpers have to be involved. Here, we don't need the involvement of a T helper. That kind of makes sense, T-dependent, T-independent. T-dependent antigens tend to be complex antigens. And you may or may not remember that I said that typically proteins and nucleic acids are the more complex antigens. So they tend to be pretty much T-dependent. T-independent antigens tend to be more simple in structure. They're oftentimes repeating units of the same monomer. So they might be things like polysaccharides, some lipids can be T-independent antigens. Another thing that you need to keep in mind is that we said that in response to a T-dependent antigen, that once this B lymphocyte starts producing antibody, it can usually class switch and make different types of that antibody. What's the first class of antibody that's produced in response to antigen stimulation? IgM. Okay? So both T-dependent antigens and independent antigens will both stimulate B cells to produce IgM. But if it's a T-dependent antigen, we can get that class switch from M to G or M to A. We can get this more kind of very robust immune response. Here, we don't get any class switching. So all they're going to produce in response to T-independent antigens are class A. So what we'll do is take a look at the T-independent antigen first, because that's pretty simple. And then that probably will be a good breaking point. So when we come back Monday, we can talk about T-dependent response. Okay, so T-independent antigens stimulate B cells without T helper involvement. They involve antigens that have repeating subunits. They only produce IgM response. What those T-independent antigens do is they tend to cross-link B cell receptors. And that's what's shown right up here. So here's a B cell. and it's got many, many copies of an identical B-cell receptor. Remember, it built that B-cell receptor through the gene shuffling process. So this particular cell will respond to one antigen type. Well, here's a polysaccharide that just contains repeating subunits of the same thing over and over again. And so this epitope right here can bind to this receptor. This is the same epitope over here, so it can bind to this receptor. This is the same epitope right here, so it can bind to this receptor. So remember, all these receptors are identical in terms of their epitope binding affinity, the antigen they bind to. And since this has multiple copies of the same epitope, this molecule tends to bind to these receptors and kind of cross-link them together. Now, they don't show this here in this particular diagram. But when these receptors become cross-linked, that particular phenomena is called capping. So this T-independent antigen kind of caps those particular receptors and sticks them together. And that causes something interesting to happen. That stimulates that B cell to undergo a process called receptor-mediated endocytosis. I don't know if you remember from cell biology or not, but what happens in receptor-mediated endocytosis is if a receptor grabs some things out on the surface of the cell, that can cause the cell membrane to pinch in and pull that substance from outside the cell to inside the cell. So basically these B cells are grabbing the antigen and pulling it inside. And long story short, basically that process stimulates those B cells to start differentiating into plasma cells and secreting their antibody. And they make class M antibody, and that's all they can make. They can't class switch because class switching involves T helper cells producing cytokines to trigger that class switching. The signal's not there. And then those antibodies are going to do something to protect you. Okay, on Monday when we go through this, we're going to look at some of these additional T-independent antigen processes. And what I would strongly recommend is that if you haven't been doing it for this particular section, particularly if it's hard for you to read these particular diagrams, is that you at least print out those particular last few PowerPoint slides, or bring your laptop or your tablet or whatever you can see them on. But really we need to be able to look at these diagrams in a little bit of detail because there's some interesting receptor interaction taking place. You have to be able to kind of see where the interleukins are being produced. And so that's going to be important. That's not going to take us the whole rest of the period Monday. So Monday, we're going to start the last topic, which are applications of immunology. We're going to be talking about using antibodies in therapy and also in serological or blood testing. So I'll make sure, I don't think I've done it yet, I'll make sure I post that last set of things, study guides, PowerPoints, and lecture notes for serology. Okay? So we'll finish up third line on Monday and then get into serology.
And ours are lack of facility, but when we look further into that lack of facility, you can't have ours
Yeah.
kind of just
Right, and so I had somebody else who had that particular situation. So what I would recommend that you do is, first thing is repeat this gelatin test.
Repeat the gelatin test?
Yep, because I mean, that's what, differentiates.
Those two.
And what you can do is, there are gelatin tubes in the side drawer there.
Okay.
What you can do is you can inoculate them, and then what temperature do you incubate at normally?
37 degrees.
Okay, so put them in 37, but that's going to melt the gelatin.
Right.
But give them time to grow in there,
Mm-hmm.
and then take them out and put them in the refrigerator again. And if they hydrolyze the gelatin, they won't
They won't
solidify.
solidify. Okay.
Yeah. The other thing too that you kind of have to think about, Peyton, is that if I've got 3 other keys that are saying one thing,
Right.
and that one that's saying a different thing, sometimes you just have to kind of go with the preponderance of evidence, because what if you have a bacillus
That's mutated.
that's a mutant for that test?
Right.
And so that's one of the reasons why I ask you to document your results,
Right.
because I can look back, and if you show me the picture and you've interpreted that test properly,
Right.
then you could come up with the quote-unquote wrong genus
Mm-hmm.
and still get full credit.
Okay.
And another thing that would happen too is that when you start looking at, okay, between lactobacillus and bacillus, endospores is one difference.
Right.
Are there other differences as well in those genus descriptions?
Right.
And you compare them to your results and you go, man, I match up really well except this one test with bacillus.
And then just repeat that.
But with lactobacillus, well, and even if the result comes out, the same,
Right.
you
Then
can still
it
go in your
still be-
report, you can go, man, I really think it's bacillus because most of the tests match up except this one. If I go with lactobacillus, several tests don't match up.
Right.
So it's really more of a matter of kind of the process you go through
Right.
rather than the right answer.
Okay.
So
All right. That helps.
try not to get frustrated by that, because these darn things mutate.
Yeah, I believe that. Thank you.
Okay. So it's kind of weird to think about the fact that this is all going on in your body right now, and what your
It is.
various cells are doing trying to fight this infection you got.